Photo shows a woman, upside down with an arched back, going over a horizontal bar at a track and field event. Figure 35. An athlete's nervous system is hard at work during the planning and execution of a movement as precise as a high jump. Parts of the nervous system are involved in determining how hard to push off and when to turn, as well as controlling the muscles throughout the body that make this complicated movement possible without knocking the bar down, all in just a few seconds. Credit Stephen Pisano. Chapter outline: 35. Neurons and glial cells. 35. How neurons communicate. 35. The central nervous system. 35. The peripheral nervous system. 35. Nervous system disorders. When you're reading this book, your nervous system is performing several functions simultaneously. The visual system is processing what is seen on the page. The motor system controls the turn of the pages or click of the mouse. The prefrontal cortex maintains attention. Even fundamental functions, like breathing and regulation of body temperature, are controlled by the nervous system. A nervous system is an organism's control center. It processes sensory information from outside and inside the body and controls all behaviors, from eating to sleeping to finding a mate. Learning objectives. By the end of this section, you will be able to do the following. List and describe the functions of the structural components of a neuron. List and describe the 4 main types of neurons. Compare the functions of different types of glial cells. Nervous systems throughout the animal kingdom vary in structure and complexity, as illustrated by the variety of animals shown in Figure 35. 2. Some organisms, like sea sponges, lack a true nervous system. Others, like jellyfish, lack a true brain and instead have a system of separate but connected nerve cells, neurons, called a nerve net. Echinoderms, such as sea stars, have nerve cells that are bundled into fibers called nerves. Flatworms of the phylum Platyhelminthes have both a central nervous system, CNS, made up of a small brain and 2 nerve cords, and a peripheral nervous system, PNS, containing a system of nerves that extend throughout the body. The insect nervous system is more complex, but also fairly decentralized. It contains a brain, ventral nerve cord, and ganglia, clusters of connected neurons. These ganglia can control movements and behaviors without input from the brain. Octopi may have the most complicated of invertebrate nervous systems. They have neurons that are organized in specialized lobes and eyes that are structurally similar to vertebrate species. Illustration A shows the nerve net of a hydra, which resembles a fish net surrounding the body. The hydra is plant-like, with a trunk and tentacle-like extensions at the top of the trunk. Illustration B shows the nervous system of a sea star. A nerve ring is present in the center of the body. Radiating out from this ring into the 5 arms are radial nerves. Illustration C shows the nervous system of a planarian, or flatworm. The flatworm has centralized ganglia, or brains, around each eye in the anterior end, and 2 nerve cords that run along the sides of the body. Transverse nerves connect the nerve cords together. Illustration D shows the nervous system of a bee. The central ganglia, or brain, is located in the head. The ventral nerve cord runs along the lower part of the body. Bumps of nerve cell bodies, called peripheral ganglia, occur periodically along the nerve cord. Illustration E shows the nervous system of the octopus, which consists of a large brain located between the 2 eyes and nerves that run into the body and arms. 2 large ganglia exist in the nerves located in the body. Illustration F shows the nervous system of a human, which consists of a central nervous system composed of the brain and spinal cord, and a peripheral nervous system composed of the nerves running into the rest of the body. Figure 35. 2 nervous systems vary in structure and complexity. In A. cnidarians, nerve cells form a decentralized nerve net. In B. echinoderms, nerve cells are bundled into fibers called nerves. In animals exhibiting bilateral symmetry, such as C. planarians, neurons cluster into an anterior brain that processes information. In addition to a brain, D. arthropods have clusters of nerve cell bodies, called peripheral ganglia, located along the ventral nerve cord. Mollusks such as squid and E. octopi, which must hunt to survive, have complex brains containing millions of neurons. In F vertebrates, the brain and spinal cord comprise the central nervous system, while neurons extending into the rest of the body comprise the peripheral nervous system. Credit E, modification of work by Michael Vecchiani, Clyde F. E. Roper, and Michael J. Sweeney, NOAA. Credit F, modification of work by NIH. Compared to invertebrates, vertebrate nervous systems are more complex, centralized, and specialized. While there is great diversity among different vertebrate nervous systems, they all share a basic structure. A CNS that contains a brain and spinal cord, and a PNS made up of peripheral sensory and motor nerves. One interesting difference between the nervous systems of invertebrates and vertebrates is that the nerve cords of many invertebrates are located ventrally, whereas the vertebrate spinal cords are located dorsally. There is debate among evolutionary biologists as to whether these different nervous system plans evolved separately or whether the invertebrate body plan arrangement somehow flipped during the evolution of vertebrates. Link to learning. Watch this video of biologist Mark Kirschner discussing the flipping phenomenon of vertebrate evolution. The nervous system is made up of neurons, specialized cells that can receive and transmit chemical or electrical signals, and glia, cells that provide support functions for the neurons by playing an information processing role that is complementary to neurons. A neuron can be compared to an electrical wire. It transmits a signal from one place to another. Glia can be compared to the workers at the electric company who make sure wires go to the right places, maintain the wires, and take down wires that are broken. Although glia have been compared to workers, recent evidence suggests that they also usurp some of the signaling functions of neurons. There is great diversity in the types of neurons and glia that are present in different parts of the nervous system. There are 4 major types of neurons, and they share several important cellular components. Neurons, the nervous system of the common laboratory fly, Drosophila melanogaster, contains around 100,000 neurons, the same number as a lobster. This number compares to 75 million in the mouse and 300 million in the octopus. A human brain contains around 86 billion neurons. Despite these very different numbers, the nervous systems of these animals control many of the same behaviors, from basic reflexes to more complicated behaviors like finding food and courting mates. The ability of neurons to communicate with each other, as well as with other types of cells, underlies all of these behaviors. Most neurons share the same cellular components, but neurons are also highly specialized. Different types of neurons have different sizes and shapes that relate to their functional roles. Parts of a neuron. Like other cells, each neuron has a cell body, or soma, that contains a nucleus, smooth and rough endoplasmic reticulum, Golgi apparatus, mitochondria, and other cellular components. Neurons also contain unique structures, illustrated in Figure 35. 3. For receiving and sending the electrical signals that make neuronal communication possible. Dendrites are tree-like structures that extend away from the cell body to receive messages from other neurons at specialized junctions called synapses. Although some neurons do not have any dendrites, some types of neurons have multiple dendrites. Dendrites can have small protrusions called dendritic spines, which further increase surface area for possible synaptic connections. Once a signal is received by the dendrite, it then travels passively to the cell body. The cell body contains a specialized structure, the axon hillock, that integrates signals from multiple synapses and serves as a junction between the cell body and an axon. An axon is a tube-like structure that propagates the integrated signal to specialized endings called axon terminals. These terminals in turn synapse on other neurons, muscle, or target organs. Chemicals released at axon terminals allow signals to be communicated to these other cells. Neurons usually have 1 or 2 axons, but some neurons, like amacrine cells in the retina, do not contain any axons. Some axons are covered with myelin, which acts as an insulator to minimize dissipation of the electrical signal as it travels down the axon, greatly increasing the speed of conduction. This insulation is important as the axon from a human motor neuron can be as long as a meter, from the base of the spine to the toes. The myelin sheath is not actually part of the neuron. Myelin is produced by glial cells. Along the axon, there are periodic gaps in the myelin sheath. These gaps are called nodes of Ranvier and are sites where the signal is recharged as it travels along the axon. It is important to note that a single neuron does not act alone. Neuronal communication depends on the connections that neurons make with one another, as well as with other cells, like muscle cells. Dendrites from a single neuron may receive synaptic contact from many other neurons. For example, dendrites from a Purkinje cell in the cerebellum are thought to receive contact from as many as 200,000 other neurons. Visual connection. Illustration shows a neuron. The main part of the cell body, called the soma, contains the nucleus. Branch-like dendrites project from 3 sides of the soma. A long, thin axon projects from the 4th side. The axon branches at the end. The tip of the axon is in close proximity to dendrites of an adjacent nerve cell. The narrow space between the axon and dendrites is called the synapse. Cells called oligodendrocytes are located next to the axon. Projections from the oligodendrocytes wrap around the axon, forming a myelin sheath. The myelin sheath is not continuous, and gaps where the axon is exposed are called nodes of Ranvier. Figure 35. 3 neurons contain organelles common to many other cells, such as a nucleus and mitochondria. They also have more specialized structures, including dendrites and axons. Which of the following statements is false? The soma is the cell body of a nerve cell. Myelin sheath provides an insulating layer to the dendrites. Axons carry the signal from the soma to the target. Dendrites carry the signal to the soma. Types of neurons. There are different types of neurons, and the functional role of a given neuron is intimately dependent on its structure. There is an amazing diversity of neuron shapes and sizes found in different parts of the nervous system and across species, as illustrated by the neurons shown in Figure 35. 4. Part A shows a pyramidal cell with 2 long, branched projections on either end of the soma. Dendrites branch from either side. Part B shows a Purkinje cell with highly branched dendrites opposite the axon. Part C shows cells with long, thin axons. The dendrites are less branched than in pyramidal or Purkinje cells. Figure 35. 4. There is great diversity in the size and shape of neurons throughout the nervous system. Examples include A. A pyramidal cell from the cerebral cortex, B. A Purkinje cell from the cerebellar cortex, and C. Olfactory cells from the olfactory epithelium and olfactory bulb. While there are many defined neuron cell subtypes, neurons are broadly divided into 4 basic types: unipolar, bipolar, multipolar, and pseudounipolar. Figure 35. 5 illustrates these 4 basic neuron types. Unipolar neurons have only one structure that extends away from the soma. These neurons are not found in vertebrates, but are found in insects where they stimulate muscles or glands. A bipolar neuron has 1 axon and 1 dendrite extending from the soma. An example of a bipolar neuron is a retinal bipolar cell, which receives signals from photoreceptor cells that are sensitive to light and transmits these signals to ganglion cells that carry the signal to the brain. Multipolar neurons are the most common type of neuron. Each multipolar neuron contains 1 axon and multiple dendrites. Multipolar neurons can be found in the central nervous system, brain, and spinal cord. An example of a multipolar neuron is a Purkinje cell in the cerebellum, which has many branching dendrites but only 1 axon. Pseudounipolar cells share characteristics with both unipolar and bipolar cells. A pseudounipolar cell has a single process that extends from the soma, like a unipolar cell, but this process later branches into 2 distinct structures, like a bipolar cell. Most sensory neurons are pseudounipolar and have an axon that branches into 2 extensions. One connected to dendrites that receive sensory information and another that transmits this information to the spinal cord. The unipolar cell has a single, long axon extending from the cell body. The bipolar neuron has 1 axon and 1 dendrite projecting from opposite sides of the cell body. The multipolar neuron has 1 long axon and several short, highly branched dendrites extending in all directions. The pseudounipolar neuron has 1 axon that forms 2 branches a short distance from the cell body, each of which extends in a different direction. Figure 35. 5 neurons are broadly divided into 4 main types based on the number and placement of axons: 1 unipolar, 2 bipolar, 3. Multipolar, and 4. Pseudounipolar. Everyday connection. Neurogenesis. At one time, scientists believed that people were born with all the neurons they would ever have. Research performed during the last few decades indicates that neurogenesis, the birth of new neurons, continues into adulthood. Neurogenesis was first discovered in songbirds that produce new neurons while learning songs. For mammals, new neurons also play an important role in learning. About 1,000 new neurons develop in the hippocampus, a brain structure involved in learning and memory, each day. While most of the new neurons will die, researchers found that an increase in the number of surviving new neurons in the hippocampus correlated with how well rats learned a new task. Interestingly, both exercise and some antidepressant medications also promote neurogenesis in the hippocampus. Stress has the opposite effect. While neurogenesis is quite limited compared to regeneration in other tissues, research in this area may lead to new treatments for disorders such as Alzheimer's, stroke, and epilepsy. How do scientists identify new neurons? A researcher can inject a compound called bromodoxyuridine, BRDU, into the brain of an animal. While all cells will be exposed to BRDU, BRDU will only be incorporated into the DNA of newly generated cells that are in S phase. A technique called immunohistochemistry can be used to attach a fluorescent label to the incorporated BRDU, and a researcher can use fluorescent microscopy to visualize the presence of BRDU, and thus new neurons, in brain tissue. Figure 35.6 is a micrograph which shows fluorescently labeled neurons in the hippocampus of a rat. In the micrograph, several cells are fluorescently shown as green only. 3 cells are shown as red only, and 4 cells are shown as being both green and red. The cells shown as green and red are astrocytes, and the cells shown red are neurons. The neurons are oval and about 10 microns long. Astrocytes are slightly larger and irregularly shaped. Figure 35.6. This micrograph shows fluorescently labeled new neurons in a rat hippocampus. Cells that are actively dividing have bromodoxyuridine, BRDU, incorporated into their DNA and are labeled in red. Cells that express glial fibrillary acidic protein, GFAP, are labeled in green. Astrocytes, but not neurons, express GFAP. Thus, cells that are labeled both red and green are actively dividing astrocytes, whereas cells labeled red only are actively dividing neurons. Credit, modification of work by Dr. Mariam Faiz et al., University of Barcelona. Scale bar data from Matt Russell. Link to learning. This video contains more information about neurogenesis. Glia. While glia are often thought of as the supporting cast of the nervous system, the number of glial cells in the brain actually outnumbers the number of neurons by a factor of 10. Neurons would be unable to function without the vital roles that are fulfilled by these glial cells. Glia guide developing neurons to their destinations, buffer ions and chemicals that would otherwise harm neurons, and provide myelin sheaths around axons. Scientists have recently discovered that they also play a role in responding to nerve activity and modulating communication between nerve cells. When glia do not function properly, the result can be disastrous. Most brain tumors are caused by mutations in glia. Types of glia. There are several different types of glia with different functions, 2 of which are shown in Figure 35.7. Astrocytes, shown in Figure 35.8a, make contact with both capillaries and neurons in the CNS. They provide nutrients and other substances to neurons, regulate the concentrations of ions and chemicals in the extracellular fluid, and provide structural support for synapses. Astrocytes also form the blood-brain barrier, a structure that blocks entrance of toxic substances into the brain. Astrocytes, in particular, have been shown through calcium imaging experiments to become active in response to nerve activity, transmit calcium waves between astrocytes, and modulate the activity of surrounding synapses. Satellite glia provide nutrients and structural support for neurons in the PNS. Microglia scavenge and degrade dead cells and protect the brain from invading microorganisms. Oligodendrocytes, shown in Figure 35.8b, form myelin sheaths around axons in the CNS. One axon can be myelinated by several oligodendrocytes, and one oligodendrocyte can provide myelin for multiple neurons. This is distinctive from the PNS, where a single Schwann cell provides myelin for only one axon as the entire Schwann cell surrounds the axon. Radial glia serve as scaffolds for developing neurons as they migrate to their end destinations. Ependymal cells line fluid-filled ventricles of the brain and the central canal of the spinal cord. They are involved in the production of cerebrospinal fluid, which serves as a cushion for the brain, moves the fluid between the spinal cord and the brain, and is a component for the choroid plexus. Illustration A shows various types of glial cells surrounding a multipolar nerve of the central nervous system. Oligodendrocytes have an oval body and protrusions that wrap around the axon. Astrocytes are round and slightly larger than neurons, with many extensions projecting outward to neurons and other cells. Microglia are small and rectangular, with many fine projections. Ependymal cells have small, round bodies lined up in a row. Long extensions connect from the ependymal cells to an astrocyte. Illustration B shows a pseudounipolar cell of the peripheral nervous system. Schwann cells wrap around the branched axon, and satellite cells surround the neuron cell body. Figure 35. 7 glial cells support neurons and maintain their environment. Glial cells of the central nervous system include oligodendrocytes, astrocytes, ependymal cells, and microglial cells. Oligodendrocytes form the myelin sheath around axons. Astrocytes provide nutrients to neurons, maintain their extracellular environment, and provide structural support. Microglia scavenge pathogens and dead cells. Ependymal cells produce cerebrospinal fluid that cushions the neurons. Glial cells of the B peripheral nervous system include Schwann cells, which form the myelin sheath, and satellite cells, which provide nutrients and structural support to neurons. Astrocytes, fluorescently labeled green, are irregularly shaped with long extensions that provide support to nerve cells. Oligodendrocytes, also labeled green, are round with long, branched extensions that form the myelin sheath of nerve cells. Figure 35. 8A astrocytes and B oligodendrocytes are glial cells of the central nervous system. Credit, a modification of work by Uniformed Services University. Credit B. Modification of work by Jurgen Broek. Scale bar data from Matt Russell. Learning objectives. By the end of this section, you will be able to do the following. Describe the basis of the resting membrane potential. Explain the stages of an action potential and how action potentials are propagated. Explain the similarities and differences between chemical and electrical synapses. Describe long-term potentiation and long-term depression. All functions performed by the nervous system, from a simple motor reflex to more advanced functions like making a memory or a decision, require neurons to communicate with one another. While humans use words and body language to communicate, neurons use electrical and chemical signals. Just like a person in a committee, one neuron usually receives and synthesizes messages from multiple other neurons before making the decision to send the message on to other neurons. Nerve impulse transmission within a neuron. For the nervous system to function, neurons must be able to send and receive signals. These signals are possible because each neuron has a charged cellular membrane, a voltage difference between the inside and the outside, and the charge of this membrane can change in response to neurotransmitter molecules released from other neurons and environmental stimuli. To understand how neurons communicate, one must first understand the basis of the baseline or resting membrane charge. Neuronal charged membranes. The lipid bilayer membrane that surrounds a neuron is impermeable to charged molecules or ions. To enter or exit the neuron, ions must pass through special proteins called ion channels that span the membrane. Ion channels have different configurations: open, closed, and inactive, as illustrated in Figure 35. 9. Some ion channels need to be activated in order to open and allow ions to pass into or out of the cell. These ion channels are sensitive to the environment and can change their shape accordingly. Ion channels that change their structure in response to voltage changes are called voltage-gated ion channels. Voltage-gated ion channels regulate the relative concentrations of different ions inside and outside the cell. The difference in total charge between the inside and outside of the cell is called the membrane potential. The first image shows a voltage-gated sodium channel that is closed at the resting potential. In response to a nerve impulse, the channel opens, allowing sodium to enter the cell. After the impulse, the channel enters an inactive state. The channel closes by a different mechanism and, for a brief period, does not reopen in response to a new nerve impulse. Figure 35. 9 voltage-gated ion channels open in response to changes in membrane voltage. After activation, they become inactivated for a brief period and will no longer open in response to a signal. Link to learning. This video discusses the basis of the resting membrane potential. Resting membrane potential. A neuron at rest is negatively charged. The inside of a cell is approximately 70 millivolts more negative than the outside, minus 70 MV. Note that this number varies by neuron type and by species. This voltage is called the resting membrane potential. It is caused by differences in the concentrations of ions inside and outside the cell. If the membrane were equally permeable to all ions, each type of ion would flow across the membrane and the system would reach equilibrium. Because ions cannot simply cross the membrane at will, there are different concentrations of several ions inside and outside the cell, as shown in Table 35. 1. The difference in the number of positively charged potassium ions, K+, inside and outside the cell dominates the resting membrane potential, Figure 35. 10. When the membrane is at rest, K+ ions accumulate inside the cell due to the activity of the Na-K pump, driving both ions against their concentration gradient. The negative resting membrane potential is created and maintained by increasing the concentration of cations outside the cell, in the extracellular fluid, relative to inside the cell, in the cytoplasm. The negative charge within the cell is created by the cell membrane being more permeable to potassium ion movement than sodium ion movement. In neurons, potassium ions are maintained at high concentrations within the cell, while sodium ions are maintained at high concentrations outside of the cell. The cell possesses potassium and sodium leakage channels that allow the two cations to diffuse down their concentration gradient. However, the neurons have far more potassium leakage channels than sodium leakage channels. Therefore, potassium diffuses out of the cell at a much faster rate than sodium leaks in. Because more cations are leaving the cell than are entering, this causes the interior of the cell to be negatively charged relative to the outside of the cell. The actions of the sodium-potassium pump help to maintain the resting potential, once established. Recall that sodium-potassium pumps brings 2 K+ ions into the cell while removing 3 Na+ ions per ATP consumed. As more cations are expelled from the cell than taken in, the inside of the cell remains negatively charged relative to the extracellular fluid. It should be noted that chloride ions, Cl, tend to accumulate outside of the cell because they are repelled by negatively charged proteins within the cytoplasm. Ion concentration inside and outside neurons. Ion extracellular concentration, intracellular concentration, ratio outside-inside. Na plus 140, 5, 12, 12. K plus 4, 155, 0. 26. Cl minus 124, 30. Organic anions a minus 100. Table 35. 1. The resting membrane potential is a result of different concentrations inside and outside the cell. The resting membrane potential of minus 70 volts is maintained by a sodium-potassium transporter that transports sodium ions out of the cell and potassium ions in. Voltage-gated sodium and potassium channels are closed. In response to a nerve impulse, some sodium channels open, allowing sodium ions to enter the cell. The membrane starts to depolarize. In other words, the charge across the membrane lessens. If the membrane potential increases to the threshold of excitation, all the sodium channels open. At the peak action potential, potassium channels open and potassium ions leave the cell. The membrane eventually becomes hyperpolarized. Figure 35. 10. The arresting membrane potential is a result of different concentrations of Na+ and K+ ions inside and outside the cell. A nerve impulse causes Na+ to enter the cell, resulting in B depolarization. At the peak action potential, K plus channels open and the cell becomes C, hyperpolarized. Action potential. A neuron can receive input from other neurons, and if this input is strong enough, send the signal to downstream neurons. Transmission of a signal between neurons is generally carried by a chemical called a neurotransmitter. Transmission of a signal within a neuron, from dendrite to axon terminal, is carried by a brief reversal of the resting membrane potential called an action potential. When neurotransmitter molecules bind to receptors located on a neuron's dendrites, ion channels open. At excitatory synapses, this opening allows positive ions to enter the neuron and results in depolarization of the membrane, a decrease in the difference in voltage between the inside and outside of the neuron. A stimulus from a sensory cell or another neuron depolarizes the target neuron to its threshold potential, MV. Na plus channels in the axon hillock open, allowing positive ions to enter the cell, figure 35.10 and figure 35.11. Once the sodium channels open, the neuron completely depolarizes to a membrane potential of about plus 40 MV. Action potentials are considered an all-or-nothing event, in that, once the threshold potential is reached, the neuron always completely depolarizes. Once depolarization is complete, the cell must now reset its membrane voltage back to the resting potential. To accomplish this, the Na plus channels close and cannot be opened. This begins the neuron's refractory period, in which it cannot produce another action potential because its sodium channels will not open. At the same time, voltage-gated K plus channels open, allowing K plus to leave the cell. As K plus ions leave the cell, the membrane potential once again becomes negative and repolarizes. The diffusion of K plus out of the cell actually continues for a short period of time past the time of the achievement of the resting potential. And the membrane hyperpolarizes, in that the membrane potential becomes more negative than the cell's normal resting potential. This is the result of the slow closing of the K plus channels. At this point, the sodium channels will return to their resting state, meaning they are ready to open again if the membrane potential again exceeds the threshold potential. Eventually, all the K plus channels close, and the cell returns back to its resting membrane potential. Visual connection. Graph plots membrane potential in millivolts versus time. The membrane remains at the resting potential of negative 70 millivolts until a nerve impulse occurs in step 1. Some sodium channels open, and the potential begins to rapidly climb past the threshold of excitation of negative 55 millivolts, at which point all the sodium channels open. At the peak action potential, the potential begins to rapidly drop as potassium channels open and sodium channels close. As a result, the membrane repolarizes past the resting membrane potential and becomes hyperpolarized. The membrane potential then gradually returns to normal. Figure 35.11. The formation of an action potential can be divided into 5 steps. 1. A stimulus from a sensory cell or another neuron causes the target cell to depolarize toward the threshold potential. 2. If the threshold of excitation is reached, all Na plus channels open and the membrane depolarizes. 3. At the peak action potential, K plus channels open and K plus begins to leave the cell. At the same time, Na plus channels close. 4. The membrane becomes hyperpolarized as K plus ions continue to leave the cell. The hyperpolarized membrane is in a refractory period and cannot fire. 5. The K plus channels close and the Na plus K plus transporter restores the resting potential. Potassium channel blockers, such as amiodarone and procainamide, which are used to treat abnormal electrical activity in the heart, called cardiac dysrhythmia, impede the movement of K plus through voltage-gated K plus channels. Which part of the action potential would you expect potassium channels to affect? In image A, the action potential travels from the soma down the axon to the axon terminal. The action potential is initiated when a signal from the soma causes the soma end of the axon membrane to depolarize. In image B, the depolarization spreads down the axon. Meanwhile, the membrane at the start of the axon repolarizes. Because potassium channels are open, the membrane cannot depolarize again. In image C, the action potential continues to spread down the axon this way. Figure 35. 12. The action potential is conducted down the axon as the axon membrane depolarizes, then repolarizes. Link to learning. This video presents an overview of action potential. Myelin and the propagation of the action potential. For an action potential to communicate information to another neuron, it must travel along the axon and reach the axon terminals, where it can initiate neurotransmitter release. The speed of conduction of an action potential along an axon is influenced by both the diameter of the axon and the axon's resistance to current leak. Myelin acts as an insulator that prevents current from leaving the axon. This increases the speed of action potential conduction. In demyelinating diseases like multiple sclerosis, action potential conduction slows because current leaks from previously insulated axon areas. The nodes of Ranvier, illustrated in Figure 35. 13 are gaps in the myelin sheath along the axon. These unmyelinated spaces are about 1 micrometer long and contain voltage-gated Na+ and K+ channels. Flow of ions through these channels, particularly the Na+ channels, regenerates the action potential over and over again along the axon. This jumping of the action potential from one node to the next is called saltatory conduction. If nodes of Ranvier were not present along an axon, the action potential would propagate very slowly, since Na+ and K+ channels would have to continuously regenerate action potentials at every point along the axon, instead of at specific points. Nodes of Ranvier also save energy for the neuron, since the channels only need to be present at the nodes and not along the entire axon. Illustration shows an axon covered in 3 bands of myelin sheath. Between the sheath coverings, the axon is exposed. The uncovered parts of the axon are called nodes of Ranvier. In the illustration, the left node of Ranvier is depolarized such that the membrane potential is positive inside and negative outside. The right membrane of the right node is at the resting potential, negative inside and positive outside. An arrow indicates that the depolarization jumps from the left node to the right, so that the right node becomes depolarized. Figure 35. 13 nodes of Ranvier are gaps in myelin coverage along axons. Nodes contain voltage-gated K+ and Na+ channels. Action potentials travel down the axon by jumping from one node to the next. Synaptic transmission. The synapse, or gap, is the place where information is transmitted from one neuron to another. Synapses usually form between axon terminals and dendritic spines, but this is not universally true. There are also axon-to-axon, dendrite-to-dendrite, and axon-to-cell body synapses. The neuron transmitting the signal is called the presynaptic neuron, and the neuron receiving the signal is called the postsynaptic neuron. Note that these designations are relative to a particular synapse. Most neurons are both presynaptic and postsynaptic. There are 2 types of synapses, chemical and electrical. Chemical synapse, when an action potential reaches the axon terminal, it depolarizes the membrane and opens voltage-gated Na+ channels. Na+ ions enter the cell, further depolarizing the presynaptic membrane. This depolarization causes voltage-gated Ca2+ channels to open. Calcium ions entering the cell initiate a signaling cascade that causes small membrane-bound vesicles, called synaptic vesicles, containing neurotransmitter molecules to fuse with the presynaptic membrane. Synaptic vesicles are shown in Figure 35. 14, which is an image from a scanning electron microscope. The axon terminal is spherical. A section is sliced off, revealing small blue and orange vesicles just inside. Figure 35. 14. This pseudo-colored image taken with a scanning electron microscope shows an axon terminal that was broken open to reveal synaptic vesicles, blue and orange, inside the neuron. Credit: modification of work by Tina Carvalho and HNIGMS. Scalebar data from Matt Russell. Fusion of a vesicle with the presynaptic membrane causes neurotransmitter to be released into the synaptic cleft. The extracellular space between the presynaptic and postsynaptic membranes, as illustrated in Figure 35. 15. The neurotransmitter diffuses across the synaptic cleft and binds to receptor proteins on the postsynaptic membrane. Illustration shows a narrow axon of a presynaptic cell widening into a bulb-like axon terminal. A narrow synaptic cleft separates the axon terminal of the presynaptic cell from the postsynaptic cell. In step 1, an action potential arrives at the axon terminal. In step 2, the action potential causes voltage-gated calcium channels in the axon terminal to open, allowing calcium to enter. In step 3, calcium influx causes neurotransmitter-containing synaptic vesicles to fuse with the plasma membrane. Contents of the vesicles are released into the synaptic cleft by exocytosis. In step 4, neurotransmitter diffuses across the synaptic cleft and binds ligand-gated ion channels on the postsynaptic membrane, causing the channels to open. In step 5, the open channels cause ion movement into or out of the cell, resulting in a localized change in membrane potential. In step 6, reuptake by the presynaptic neuron, enzymatic degradation and diffusion reduce neurotransmitter levels, terminating the signal. Figure 35. 15. Communication at chemical synapses requires release of neurotransmitters. When the presynaptic membrane is depolarized, voltage-gated Ca2 plus channels open and allow Ca2 plus to enter the cell. The calcium entry causes synaptic vesicles to fuse with the membrane and release neurotransmitter molecules into the synaptic cleft. The neurotransmitter diffuses across the synaptic cleft and binds to ligand-gated ion channels in the postsynaptic membrane, resulting in a localized depolarization or hyperpolarization of the postsynaptic neuron. The binding of a specific neurotransmitter causes particular ion channels, in this case ligand-gated channels, on the postsynaptic membrane to open. Neurotransmitters can either have excitatory or inhibitory effects on the postsynaptic membrane. For example, when acetylcholine is released at the synapse between a nerve and muscle, called the neuromuscular junction, by a presynaptic neuron, it causes postsynaptic Na+ channels to open. Na+ enters the postsynaptic cell and causes the postsynaptic membrane to depolarize. This depolarization is called an excitatory postsynaptic potential, EPSP, and makes the postsynaptic neuron more likely to fire an action potential. Release of neurotransmitter at inhibitory synapses causes inhibitory postsynaptic potentials, IPSPs, a hyperpolarization of the presynaptic membrane. For example, when the neurotransmitter GABA, gamma-aminobutyric acid, is released from a presynaptic neuron, it binds to and opens Cl channels. Cl ions enter the cell and hyperpolarizes the membrane, making the neuron less likely to fire an action potential. Once neurotransmission has occurred, the neurotransmitter must be removed from the synaptic cleft so the postsynaptic membrane can reset and be ready to receive another signal. This can be accomplished in 3 ways. The neurotransmitter can diffuse away from the synaptic cleft, it can be degraded by enzymes in the synaptic cleft, or it can be recycled, sometimes called reuptake, by the presynaptic neuron. Several drugs act at this step of neurotransmission. For example, some drugs that are given to Alzheimer's patients work by inhibiting acetylcholinesterase, the enzyme that degrades acetylcholine. This inhibition of the enzyme essentially increases neurotransmission at synapses that release acetylcholine. Once released, the acetylcholine stays in the cleft and can continually bind and unbind to postsynaptic receptors. Neurotransmitter function and location. Neurotransmitter, example location, acetylcholine, CNS and/or PNS, biogenic amine dopamine, serotonin, norepinephrine CNS and/or PNS, amino acid glycine, glutamate, aspartate, gamma-aminobutyric acid CNS, neuropeptide substance P, endorphin CNS and/or PNS. Table 35. 2. Electrical synapse. While electrical synapses are fewer in number than chemical synapses, they are found in all nervous systems and play important and unique roles. The mode of neurotransmission in electrical synapses is quite different from that in chemical synapses. In an electrical synapse, the presynaptic and postsynaptic membranes are very close together and are actually physically connected by channel proteins forming gap junctions. Gap junctions allow current to pass directly from one cell to the next. In addition to the ions that carry this current, other molecules, such as ATP, can diffuse through the large gap junction pores. There are key differences between chemical and electrical synapses. Because chemical synapses depend on the release of neurotransmitter molecules from synaptic vesicles to pass on their signal, there is an approximately 1 millisecond delay between when the axon potential reaches the presynaptic terminal and when the neurotransmitter leads to opening of postsynaptic ion channels. Additionally, this signaling is unidirectional. Signaling in electrical synapses, in contrast, is virtually instantaneous, which is important for synapses involved in key reflexes, and some electrical synapses are bidirectional. Electrical synapses are also more reliable as they are less likely to be blocked, and they are important for synchronizing the electrical activity of a group of neurons. For example, electrical synapses in the thalamus are thought to regulate slow-wave sleep, and disruption of these synapses can cause seizures. Signal summation. Sometimes, a single EPSB is strong enough to induce an action potential in the postsynaptic neuron, but often multiple presynaptic inputs must create EPSBs around the same time for the postsynaptic neuron to be sufficiently depolarized to fire an action potential. This process is called summation and occurs at the axon hillock, as illustrated in Figure 35. 16. Additionally, one neuron often has inputs from many presynaptic neurons, some excitatory and some inhibitory, so IPSBs can cancel out EPSBs and vice versa. It is the net change in postsynaptic membrane voltage that determines whether the postsynaptic cell has reached its threshold of excitation needed to fire an action potential. Together, synaptic summation and the threshold for excitation act as a filter so that random noise in the system is not transmitted as important information. Illustration shows the location of the axon hillock, which is the area connecting the neuron body to the axon. A graph shows the summation of membrane potentials at the axon hillock, plotted as membrane potential in millivolts versus time. Initially, the membrane potential at the axon hillock is minus 70 millivolts. A series of EPSPS and IPSPS cause the potential to rise and fall. Eventually, the potential increases to the threshold of excitation. At this point, the nerve fires, resulting in a sharp increase in membrane potential, followed by a rapid decrease. The hillock becomes hyperpolarized such that the membrane potential is lower than the resting potential. The hillock then gradually returns to the resting potential. Figure 35. 16. A single neuron can receive both excitatory and inhibitory inputs from multiple neurons, resulting in local membrane depolarization, EPSB input, and hyperpolarization, IPSB input. All these inputs are added together at the axon hillock. If the EPSBs are strong enough to overcome the IPSBs and reach the threshold of excitation, the neuron will fire. Everyday connection, brain-computer interface, amyotrophic lateral sclerosis, ALS, also called Lou Gehrig's disease, is a neurological disease characterized by the degeneration of the motor neurons that control voluntary movements. The disease begins with muscle weakening and lack of coordination, and eventually destroys the neurons that control speech, breathing, and swallowing. In the end, the disease can lead to paralysis. At that point, patients require assistance from machines to be able to breathe and to communicate. Several special technologies have been developed to allow, locked-in, patients to communicate with the rest of the world. One technology, for example, allows patients to type out sentences by twitching their cheek. These sentences can then be read aloud by a computer. A relatively new line of research for helping paralyzed patients, including those with ALS, to communicate and retain a degree of self-sufficiency is called brain-computer interface, BCI technology, and is illustrated in Figure 35. 17. This technology sounds like something out of science fiction. It allows paralyzed patients to control a computer using only their thoughts. There are several forms of BCI. Some forms use EEG recordings from electrodes taped onto the skull. These recordings contain information from large populations of neurons that can be decoded by a computer. Other forms of BCI require the implantation of an array of electrodes smaller than a postage stamp in the arm and hand area of the motor cortex. This form of BCI, while more invasive, is very powerful as each electrode can record actual action potentials from one or more neurons. These signals are then sent to a computer, which has been trained to decode the signal and feed it to a tool, such as a cursor on a computer screen. This means that a patient with ALS can use email, read the internet, and communicate with others by thinking of moving their hand or arm, even though the paralyzed patient cannot make that bodily movement. Recent advances have allowed a paralyzed locked-in patient who suffered a stroke 15 years ago to control a robotic arm and even to feed herself coffee using BCI technology. Despite the amazing advancements in BCI technology, it also has limitations. The technology can require many hours of training and long periods of intense concentration for the patient. It can also require brain surgery to implant the devices. Illustration shows a person in a wheelchair facing a computer screen. An arrow indicates that neural signals travel from the brain of the paralyzed person to the computer. Figure 35. 17. With brain-computer interface technology, neural signals from a paralyzed patient are collected, decoded, and then fed to a tool, such as a computer, a wheelchair, or a robotic arm. Link to learning. Watch this video in which a paralyzed woman uses a brain-controlled robotic arm to bring a drink to her mouth, among other images of brain-computer interface technology in action. Synaptic plasticity. Synapses are not static structures. They can be weakened or strengthened. They can be broken, and new synapses can be made. Synaptic plasticity allows for these changes, which are all needed for a functioning nervous system. In fact, synaptic plasticity is the basis of learning and memory. Two processes in particular, long-term potentiation, LTP, and long-term depression, LTD, are important forms of synaptic plasticity that occur in synapses in the hippocampus, a brain region that is involved in storing memories. Long-term potentiation, LTP. Long-term potentiation, LTP, is a persistent strengthening of a synaptic connection. LTP is based on the Hebbian principle. Cells that fire together wire together. There are various mechanisms, none fully understood, behind the synaptic strengthening scene with LTP. One known mechanism involves a type of postsynaptic glutamate receptor, called NMDA and methyldiaspartate receptors, shown in Figure 35. 18. These receptors are normally blocked by magnesium ions. However, when the postsynaptic neuron is depolarized by multiple presynaptic inputs in quick succession, either from one neuron or multiple neurons, the magnesium ions are forced out, allowing Ca2+ ions to pass into the postsynaptic cell. Next, Ca2+ ions entering the cell initiate a signaling cascade that causes a different type of glutamate receptor, called AMPA, alpha-amino 3-hydroxy-5-methyl-4-isoxazole-propionic acid, receptors, to be inserted into the postsynaptic membrane, since activated AMPA receptors allow positive ions to enter the cell. So, the next time glutamate is released from the presynaptic membrane, it will have a larger excitatory effect, EPSB, on the postsynaptic cell, because the binding of glutamate to these AMPA receptors will allow more positive ions into the cell. The insertion of additional AMPA receptors strengthens the synapse and means that the postsynaptic neuron is more likely to fire in response to presynaptic neurotransmitter release. Some drugs of abuse co-opt the LTP pathway, and this synaptic strengthening can lead to addiction. Long-term depression, LTD. Long-term depression, LTD, is essentially the reverse of LTP. It is a long-term weakening of a synaptic connection. One mechanism known to cause LTD also involves AMPA receptors. In this situation, calcium that enters through NMDA receptors initiates a different signaling cascade, which results in the removal of AMPA receptors from the postsynaptic membrane, as illustrated in Figure 35. 18. The decrease in AMPA receptors in the membrane makes the postsynaptic neuron less responsive to glutamate released from the presynaptic neuron. While it may seem counterintuitive, LTD may be just as important for learning and memory as LTP. The weakening and pruning of unused synapses allows for unimportant connections to be lost, and makes the synapses that have undergone LTP that much stronger by comparison. Illustration shows the mechanism of LTP and LTD normally. The NMDA receptor in the postsynaptic neuron is activated by glutamate binding, but only after depolarization removes an inhibitory magnesium ion. Once the magnesium is removed, calcium can enter the cell. In response to an increase in intracellular calcium, AMPA receptors are inserted into the plasma membrane, which amplifies the signal resulting in LTP. LDP occurs when low-frequency stimulation results in the activation of a different calcium signaling cascade that causes AMPA receptors to be removed from the plasma membrane. As a result, the nerve cell becomes less responsive to glutamate. Figure 35. 18 calcium entry through postsynaptic NMDA receptors can initiate two different forms of synaptic plasticity: long-term potentiation, LTP, and long-term depression, LTD. LTP arises when a single synapse is repeatedly stimulated. This stimulation causes a calcium and Ka-MKI-dependent cellular cascade, which results in the insertion of more AMPA receptors into the postsynaptic membrane. The next time glutamate is released from the presynaptic cell, it will bind to both NMDA and the newly inserted AMPA receptors, thus depolarizing the membrane more efficiently. LTD occurs when few glutamate molecules bind to NMDA receptors at a synapse, due to a low firing rate of the presynaptic neuron. The calcium that does flow through NMDA receptors initiates a different calcineurin and protein phosphatase 1-dependent cascade, which results in the endocytosis of AMPA receptors. This makes the postsynaptic neuron less responsive to glutamate released from the presynaptic neuron. Learning objectives. By the end of this section, you will be able to do the following: identify the spinal cord, cerebral lobes, and other brain areas on a diagram of the brain. Describe the basic functions of the spinal cord, cerebral lobes, and other brain areas. The central nervous system, CNS, is made up of the brain, a part of which is shown in Figure 35. 19 and spinal cord, and is covered with 3 layers of protective coverings called meninges, from the Greek word for membrane. The outermost layer is the dura mater, Latin for hard mother. As the Latin suggests, the primary function for this thick layer is to protect the brain and spinal cord. The dura mater also contains vein-like structures that carry blood from the brain back to the heart. The middle layer is the web-like arachnoid mater. The last layer is the pia mater, Latin for soft mother, which directly contacts and covers the brain and spinal cord like plastic wrap. The space between the arachnoid and pia maters is filled with cerebrospinal fluid, CSF. CSF is produced by a tissue called choroid plexus in fluid-filled compartments in the CNS called ventricles. The brain floats in CSF, which acts as a cushion and shock absorber and makes the brain neutrally buoyant. CSF also functions to circulate chemical substances throughout the brain and into the spinal cord. The entire brain contains only about 5 tablespoons of CSF, but CSF is constantly produced in the ventricles. This creates a problem when a ventricle is blocked. The CSF builds up and creates swelling, and the brain is pushed against the skull. This swelling condition is called hydrocephalus, waterhead, and can cause seizures, cognitive problems, and even death if a shunt is not inserted to remove the fluid and pressure. Illustration shows the 3 meninges that protect the brain. The outermost layer, just beneath the skull, is the dura mater. The dura mater is the thickest meninge, and blood vessels run through it. Beneath the dura mater is the arachnoid mater, and beneath this is the pia mater. Figure 35. 19. The cerebral cortex is covered by 3 layers of meninges, the dura, arachnoid, and pia maters. Credit: Modification of work by Gray's Anatomy. Brain. The brain is the part of the central nervous system that is contained in the cranial cavity of the skull. It includes the cerebral cortex, limbic system, basal ganglia, thalamus, hypothalamus, and cerebellum. There are 3 different ways that a brain can be sectioned in order to view internal structures. A sagittal section cuts the brain left to right, as shown in Figure 35. 21b, a coronal section cuts the brain front to back, as shown in Figure 35. 20a, and a horizontal section cuts the brain top to bottom. Cerebral cortex. The outermost part of the brain is a thick piece of nervous system tissue called the cerebral cortex, which is folded into hills called gyri singular, gyrus, and valleys called sulci singular, sulcus. The cortex is made up of two hemispheres, right and left, which are separated by a large sulcus. A thick fiber bundle called the corpus callosum, Latin, tough body, connects the two hemispheres and allows information to be passed from one side to the other. Although there are some brain functions that are localized more to one hemisphere than the other, the functions of the two hemispheres are largely redundant. In fact, sometimes, very rarely, an entire hemisphere is removed to treat severe epilepsy. While patients do suffer some deficits following the surgery, they can have surprisingly few problems, especially when the surgery is performed on children who have very immature nervous systems. Illustration shows coronal front and sagittal side sections of a human brain. In the coronal section, the large upper part of the brain, called the cerebral cortex, is divided into left and right hemispheres. A cavity resembling butterfly wings exists between the left and right cortex. The corpus callosum is a band that connects the two hemispheres together just above this cavity. The surface of the cerebral cortex contains bumpy protrusions called gyri. The cerebral cortex is anchored by the brainstem, which connects with the spinal cord. On either side of the brainstem tucked beneath the cerebral cortex is the cerebellum. The surface of the cerebellum is bumpy, but not as bumpy as the cerebral cortex. The sagittal section reveals that the cerebral cortex makes up the front and top part of the brain, while the brainstem and cerebellum make up the lower back part. The oval thalamus sits in the cavity in the middle of the cerebral cortex. The corpus callosum wraps around the top part of the thalamus. The basal ganglia wraps around the corpus callosum, starting at the lower front part of the brain and continuing 3/4 of the way around so the back end almost meets the front end. The basal ganglia is separated into segments that are connected along the top and bottom. The amygdala is a spherical structure at the end of the basal ganglia. Figure 35. 20 These illustrations show the A coronal and B sagittal sections of the human brain. In other surgeries to treat severe epilepsy, the corpus callosum is cut instead of removing an entire hemisphere. This causes a condition called split brain, which gives insights into unique functions of the two hemispheres. For example, when an object is presented to patients, left visual field, they may be unable to verbally name the object and may claim to not have seen an object at all. This is because the visual input from the left visual field crosses and enters the right hemisphere and cannot then signal to the speech center, which generally is found in the left side of the brain. Remarkably, if a split brain patient is asked to pick up a specific object out of a group of objects with the left hand, the patient will be able to do so, but will still be unable to vocally identify it. Link to learning. View this video to learn more about split brain patients. Each cortical hemisphere contains regions called lobes that are involved in different functions. Scientists use various techniques to determine what brain areas are involved in different functions. They examine patients who have had injuries or diseases that affect specific areas and see how those areas are related to functional deficits. They also conduct animal studies where they stimulate brain areas and see if there are any behavioral changes. They use a technique called transcranial magnetic stimulation, TMS, to temporarily deactivate specific parts of the cortex using strong magnets placed outside the head. And they use functional magnetic resonance imaging, fMRI, to look at changes in oxygenated blood flow in particular brain regions that correlate with specific behavioral tasks. These techniques, and others, have given great insight into the functions of different brain regions, but have also showed that any given brain area can be involved in more than one behavior or process. And any given behavior or process generally involves neurons in multiple brain areas. That being said, each hemisphere of the mammalian cerebral cortex can be broken down into 4 functionally and spatially defined lobes: frontal, parietal, temporal, and occipital. Figure 35. 21 illustrates these 4 lobes of the human cerebral cortex. Sagittal, or side view of the human brain, shows the different lobes of the cerebral cortex. The frontal lobe is at the front center of the brain. The parietal lobe is at the top back part of the brain. The occipital lobe is at the back of the brain, and the temporal lobe is at the bottom center of the brain. The motor cortex is the back of the frontal lobe, and the olfactory bulb is the bottom part. The somatosensory cortex is the front part of the parietal lobe. The brainstem is beneath the temporal lobe, and the cerebellum is beneath the occipital lobe. Figure 35. 21. The human cerebral cortex includes the frontal, parietal, temporal, and occipital lobes. The frontal lobe is located at the front of the brain, over the eyes. This lobe contains the olfactory bulb, which processes smells. The frontal lobe also contains the motor cortex, which is important for planning and implementing movement. Areas within the motor cortex map to different muscle groups, and there is some organization to this map, as shown in Figure 35. 22. For example, the neurons that control movement of the fingers are next to the neurons that control movement of the hand. Neurons in the frontal lobe also control cognitive functions like maintaining attention, speech, and decision-making. Studies of humans who have damaged their frontal lobes show that parts of this area are involved in personality, socialization, and assessing risk. Diagram shows the location of motor control for various muscle groups on the right hemisphere cerebral cortex. From the top middle of the motor cortex to the bottom right, the order of areas controlled is toes, ankles, knees, hips, trunk, shoulders, elbows, wrists, hands, fingers, thumbs, neck, eyebrows and eyelids, eyeballs, face, lips, jaw, tongue, salivation, chewing, and swallowing. Figure 35. 22 different parts of the motor cortex control different muscle groups. Muscle groups that are neighbors in the body are generally controlled by neighboring regions of the motor cortex as well. For example, the neurons that control finger movement are near the neurons that control hand movement. The parietal lobe is located at the top of the brain. Neurons in the parietal lobe are involved in speech and also reading. Two of the parietal lobe's main functions are processing somatosensation, touch sensations like pressure, pain, heat, cold, and processing proprioception, the sense of how parts of the body are oriented in space. The parietal lobe contains a somatosensory map of the body similar to the motor cortex. The occipital lobe is located at the back of the brain. It is primarily involved in vision, seeing, recognizing, and identifying the visual world. The temporal lobe is located at the base of the brain by your ears and is primarily involved in processing and interpreting sounds. It also contains the hippocampus, Greek for seahorse, a structure that processes memory formation. The hippocampus is illustrated in Figure 35. 24. The role of the hippocampus in memory was partially determined by studying one famous epileptic patient, H.M., who had both sides of his hippocampus removed in an attempt to cure his epilepsy. His seizures went away, but he could no longer form new memories, although he could remember some facts from before his surgery and could learn new motor tasks. Evolution connection. Cerebral cortex. Compared to other vertebrates, mammals have exceptionally large brains for their body size. An entire alligator's brain, for example, would fill about 1.5 teaspoons. This increase in brain-to-body size ratio is especially pronounced in apes, whales, and dolphins. While this increase in overall brain size doubtlessly played a role in the evolution of complex behaviors unique to mammals, it does not tell the whole story. Scientists have found a relationship between the relatively high surface area of the cortex and the intelligence and complex social behaviors exhibited by some mammals. This increased surface area is due, in part, to increased folding of the cortical sheet, more sulci and gyri. For example, a rat cortex is very smooth with very few sulci and gyri. Cat and sheep cortices have more sulci and gyri. Chimps, humans, and dolphins have even more. Illustrations shows that brains increase in size and amount of cortical folding from rat to cat to chimpanzee to human to dolphin. Figure 35. 23 mammals have larger brain-to-body ratios than other vertebrates. Within mammals, increased cortical folding and surface area is correlated with complex behavior. Basal ganglia. Interconnected brain areas called the basal ganglia, or basal nuclei, shown in Figure 35. 20b play important roles in movement control and posture. Damage to the basal ganglia, as in Parkinson's disease, leads to motor impairments like a shuffling gait when walking. The basal ganglia also regulate motivation. For example, when a wasp sting led to bilateral basal ganglia damage in a 25-year-old businessman. He began to spend all his days in bed and showed no interest in anything or anybody. But when he was externally stimulated, as when someone asked to play a card game with him, he was able to function normally. Interestingly, he and other similar patients do not report feeling bored or frustrated by their state. Thalamus, the thalamus, Greek for inner chamber, illustrated in figure 35. 24 acts as a gateway to and from the cortex. It receives sensory and motor inputs from the body and also receives feedback from the cortex. This feedback mechanism can modulate conscious awareness of sensory and motor inputs depending on the attention and arousal state of the animal. The thalamus helps regulate consciousness, arousal, and sleep states. A rare genetic disorder called fatal familial insomnia causes the degeneration of thalamic neurons and glia. This disorder prevents affected patients from being able to sleep, among other symptoms, and is eventually fatal. Illustration shows parts of the limbic system. The thalamus and hypothalamus are located in the cavity in the center of the cerebral cortex. The cingulate gyrus is part of the cerebral cortex that wraps around the upper part of the basal ganglia. The hippocampus is part of the cerebral cortex located beneath the thalamus. The amygdala is located at the end of the basal ganglia and sits beside the pituitary. Figure 35. 24. The limbic system regulates emotion and other behaviors. It includes parts of the cerebral cortex located near the center of the brain, including the cingulate gyrus and the hippocampus, as well as the thalamus, hypothalamus, and amygdala. Hypothalamus. Below the thalamus is the hypothalamus, shown in figure 35. 24. The hypothalamus controls the endocrine system by sending signals to the pituitary gland, a pea-sized endocrine gland that releases several different hormones that affect other glands as well as other cells. This relationship means that the hypothalamus regulates important behaviors that are controlled by these hormones. The hypothalamus is the body's thermostat. It makes sure key functions like food and water intake, energy expenditure, and body temperature are kept at appropriate levels. Neurons within the hypothalamus also regulate circadian rhythms, sometimes called sleep cycles. Limbic system. The limbic system is a connected set of structures that regulates emotion, as well as behaviors related to fear and motivation. It plays a role in memory formation and includes parts of the thalamus and hypothalamus as well as the hippocampus. One important structure within the limbic system is a temporal lobe structure called the amygdala, Greek for almond, illustrated in figure 35. 24. The two amygdala are important both for the sensation of fear and for recognizing fearful faces. The cingulate gyrus helps regulate emotions and pain. Cerebellum. The cerebellum, Latin for little brain, shown in figure 35. 21, sits at the base of the brain on top of the brainstem. The cerebellum controls balance and aids in coordinating movement and learning new motor tasks. Brainstem. The brainstem, illustrated in figure 35. 21, connects the rest of the brain with the spinal cord. It consists of the midbrain, medulla oblongata, and the pons. Motor and sensory neurons extend through the brainstem, allowing for the relay of signals between the brain and spinal cord. Ascending neural pathways cross in this section of the brain, allowing the left hemisphere of the cerebrum to control the right side of the body and vice versa. The brainstem coordinates motor control signals sent from the brain to the body. The brainstem controls several important functions of the body, including alertness, arousal, breathing, blood pressure, digestion, heart rate, swallowing, walking, and sensory, and motor information integration. Spinal cord. Connecting to the brainstem and extending down the body through the spinal column is the spinal cord, shown in figure 35. 21. The spinal cord is a thick bundle of nerve tissue that carries information about the body to the brain and from the brain to the body. The spinal cord is contained within the bones of the vertebrate column, but is able to communicate signals to and from the body through its connections with spinal nerves, part of the peripheral nervous system. A cross-section of the spinal cord looks like a white oval containing a gray butterfly shape, as illustrated in figure 35. 25. Myelinated axons make up the white matter and neuron and glial cell bodies make up the gray matter. Gray matter is also composed of interneurons, which connect two neurons each located in different parts of the body. Axons and cell bodies in the dorsal, facing the back of the animal, spinal cord, convey mostly sensory information from the body to the brain. Axons and cell bodies in the ventral, facing the front of the animal, spinal cord, primarily transmit signals controlling movement from the brain to the body. The spinal cord also controls motor reflexes. These reflexes are quick, unconscious movements, like automatically removing a hand from a hot object. Reflexes are so fast because they involve local synaptic connections. For example, the knee reflex that a doctor tests during a routine physical is controlled by a single synapse between a sensory neuron and a motor neuron. While a reflex may only require the involvement of one or two synapses, synapses with interneurons in the spinal column transmit information to the brain to convey what happened, the knee jerked, or the hand was hot. In the United States, there are around 10,000 spinal cord injuries each year. Because the spinal cord is the information superhighway connecting the brain with the body, damage to the spinal cord can lead to paralysis. The extent of the paralysis depends on the location of the injury along the spinal cord and whether the spinal cord was completely severed. For example, if the spinal cord is damaged at the level of the neck, it can cause paralysis from the neck down, whereas damage to the spinal column further down may limit paralysis to the legs. Spinal cord injuries are notoriously difficult to treat because spinal nerves do not regenerate, although ongoing research suggests that stem cell transplants may be able to act as a bridge to reconnect severed nerves. Researchers are also looking at ways to prevent the inflammation that worsens nerve damage after injury. One such treatment is to pump the body with cold saline to induce hypothermia. This cooling can prevent swelling and other processes that are thought to worsen spinal cord injuries. In the cross-section, the gray matter forms an X inside the oval white matter. The legs of the X are thicker than the arms. Each leg is called a ventral horn, and each arm is called a dorsal horn. Figure 35. 25. A cross-section of the spinal cord shows gray matter, containing cell bodies and interneurons, and white matter containing axons. Learning objectives. By the end of this section, you will be able to do the following. Describe the organization and functions of the sympathetic and parasympathetic nervous systems. Describe the organization and function of the sensory-somatic nervous system. The peripheral nervous system, PNS, is the connection between the central nervous system and the rest of the body. The CNS is like the power plant of the nervous system. It creates the signals that control the functions of the body. The PNS is like the wires that go to individual houses. Without those wires, the signals produced by the CNS could not control the body, and the CNS would not be able to receive sensory information from the body either. The PNS can be broken down into the autonomic nervous system, which controls bodily functions without conscious control, and the sensory-somatic nervous system, which transmits sensory information from the skin, muscles, and sensory organs to the CNS and sends motor commands from the CNS to the muscles. Autonomic nervous system. Visual connection. The autonomic nervous system is divided into sympathetic and parasympathetic systems. In the sympathetic system, the soma of the preganglionic neurons is usually located in the spine, while in the parasympathetic system, the soma is usually in the brainstem or sacral vertebrae, at the bottom of the spine. In both systems, the preganglionic neuron releases the neurotransmitter acetylcholine into the preganglionic synapse. Postganglionic neurons of the sympathetic system have somas in a sympathetic ganglion, located next to the spinal cord. Postganglionic neurons of the parasympathetic system have somas in ganglions near the target organ. Postganglionic neurons of the sympathetic system release norepinephrine into the postganglionic synapse, causing the fight or flight response to be activated in the target organ, while postganglionic neurons of the parasympathetic system release acetylcholine or nitric oxide into postganglionic synapse, causing the rest and digest response to be activated in the target organ. Figure 35. 26. In the autonomic nervous system, a preganglionic neuron of the CNS synapses with a postganglionic neuron of the PNS. The The postganglionic neuron, in turn, acts on a target organ. Autonomic responses are mediated by the sympathetic and the parasympathetic systems, which are antagonistic to one another. The sympathetic system activates the fight or flight response, while the parasympathetic system activates the rest and digest response. Which of the following statements is false? The parasympathetic pathway is responsible for resting the body, while the sympathetic pathway is responsible for preparing for an emergency. Most preganglionic neurons in the sympathetic pathway originate in the spinal cord. Slowing of the heartbeat is a parasympathetic response. Parasympathetic neurons are responsible for releasing norepinephrine on the target organ, while sympathetic neurons are responsible for releasing acetylcholine. The autonomic nervous system serves as the relay between the CNS and the internal organs. It controls the lungs, the heart, smooth muscle, and exocrine and endocrine glands. The autonomic nervous system controls these organs largely without conscious control. It can continuously monitor the conditions of these different systems and implement changes as needed. Signaling to the target tissue usually involves two synapses: a preganglionic neuron, originating in the CNS, synapses to a neuron in a ganglion that, in turn, synapses on the target organ, as illustrated in Figure 35.26. There are two divisions of the autonomic nervous system that often have opposing effects: the sympathetic nervous system and the parasympathetic nervous system. Sympathetic nervous system. The sympathetic nervous system is responsible for the fight or flight response that occurs when an animal encounters a dangerous situation. One way to remember this is to think of the surprise a person feels when encountering a snake. Snake and sympathetic both begin with S. Examples of functions controlled by the sympathetic nervous system include an accelerated heart rate and inhibited digestion. These functions help prepare an organism's body for the physical strain required to escape a potentially dangerous situation or to fend off a predator. Illustration shows the effects of the sympathetic and parasympathetic systems on target organs and the placement of the preganglionic neurons that mediate these effects. The parasympathetic system causes pupils and bronchi to constrict, slows the heart rate, and stimulates salivation, digestion, and bile secretion. Preganglionic neurons that mediate these effects are all located in the brainstem. Preganglionic neurons of the parasympathetic system that are located in the sacral cause the bladder to contract. The sympathetic system causes pupils and bronchi to dilate, increases heart rate, inhibits digestion, stimulates the breakdown of glycogen and the secretion of adrenaline and noradrenaline, and inhibits contraction of the bladder. The preganglionic neurons that mediate these effects are all located in the spine. Figure 35.27. The sympathetic and parasympathetic nervous systems often have opposing effects on target organs. Most preganglionic neurons in the sympathetic nervous system originate in the spinal cord, as illustrated in Figure 35.27. The axons of these neurons release acetylcholine on postganglionic neurons within sympathetic ganglia. The sympathetic ganglia form a chain that extends alongside the spinal cord. The acetylcholine activates the postganglionic neurons. Postganglionic neurons then release norepinephrine onto target organs. As anyone who has ever felt a rush before a big test, speech, or athletic event can attest, the effects of the sympathetic nervous system are quite pervasive. This is both because one preganglionic neuron synapses on multiple postganglionic neurons, amplifying the effect of the original synapse, and because the adrenal gland also releases norepinephrine and the closely related hormone epinephrine into the bloodstream. The physiological effects of this norepinephrine release include dilating the trachea and bronchi, making it easier for the animal to breathe, increasing heart rate, and moving blood from the skin to the heart, muscles, and brain, so the animal can think and run. The strength and speed of the sympathetic response helps an organism avoid danger, and scientists have found evidence that it may also increase LTP, allowing the animal to remember the dangerous situation and avoid it in the future. Parasympathetic nervous system. While the sympathetic nervous system is activated in stressful situations, the parasympathetic nervous system allows an animal to rest and digest. One way to remember this is to think that during a restful situation like a picnic, the parasympathetic nervous system is in control. Picnic and parasympathetic both start with P. Parasympathetic preganglionic neurons have cell bodies located in the brainstem and in the sacral, toward the bottom, spinal cord, as shown in Figure 35. 27. The axons of the preganglionic neurons release acetylcholine on the postganglionic neurons, which are generally located very near the target organs. Most postganglionic neurons release acetylcholine onto target organs, although some release nitric oxide. The parasympathetic nervous system resets organ function after the sympathetic nervous system is activated. The common adrenaline dump you feel after a fight-or-flight event. Effects of acetylcholine release on target organs include slowing of heart rate, lowered blood pressure, and stimulation of digestion. Sensory somatic nervous system. The sensory somatic nervous system is made up of cranial and spinal nerves and contains both sensory and motor neurons. Sensory neurons transmit sensory information from the skin, skeletal muscle, and sensory organs to the CNS. Motor neurons transmit messages about desired movement from the CNS to the muscles to make them contract. Without its sensory somatic nervous system, an animal would be unable to process any information about its environment, what it sees, feels, hears, and so on, and could not control motor movements. Unlike the autonomic nervous system, which has two synapses between the CNS and the target organ, sensory and motor neurons have only one synapse. One ending of the neuron is at the organ, and the other directly contacts a CNS neuron. Acetylcholine is the main neurotransmitter released at these synapses. Humans have 12 cranial nerves, nerves that emerge from or enter the skull, cranium, as opposed to the spinal nerves, which emerge from the vertebral column. Each cranial nerve is accorded a name, which are detailed in Figure 35. 28. Some cranial nerves transmit only sensory information. For example, the olfactory nerve transmits information about smells from the nose to the brainstem. Other cranial nerves transmit almost solely motor information. For example, the oculomotor nerve controls the opening and closing of the eyelid and some eye movements. Other cranial nerves contain a mix of sensory and motor fibers. For example, the glossopharyngeal nerve has a role in both taste, sensory, and swallowing motor. Illustration shows the underside of the brain. The 12 cranial nerves cluster around the brainstem and are symmetrically located on each side. The olfactory nerve is short and lobe-like and is located closest to the front. Directly behind this is the optic nerve, then the oculomotor nerve. All these nerves are located in front of the brainstem. The trigeminal nerve, which is the thickest, is located on either side of the brainstem. It forms 3 branches shortly after leaving the brain. The trochlear nerve is a small nerve in front of the trigeminal nerve. Behind the brainstem are the smaller facial, vestibulocochlear, glossopharyngeal, and hypoglossal nerves. The nerve furthest back is the accessory nerve. Figure 35. 28. The human brain contains 12 cranial nerves that receive sensory input and control motor output for the head and neck. Spinal nerves transmit sensory and motor information between the spinal cord and the rest of the body. Each of the 31 spinal nerves, in humans, contains both sensory and motor axons. The sensory neuron cell bodies are grouped in structures called dorsal root ganglia and are shown in Figure 35. 29. Each sensory neuron has one projection, with a sensory receptor ending in skin, muscle, or sensory organs, and another that synapses with a neuron in the dorsal spinal cord. Motor neurons have cell bodies in the ventral gray matter of the spinal cord that project to muscle through the ventral root. These neurons are usually stimulated by interneurons within the spinal cord, but are sometimes directly stimulated by sensory neurons. Illustration shows a cross-section of the spinal cord. The gray matter forms an X inside the white matter. A spinal nerve extends from the left arm of the X, and another extends from the left leg of the X. The two nerves join together to the left of the spine. The right arm and leg of the X form a symmetrical nerve. The part of the nerve that exits from the leg of the X is called the ventral root, and the part that exits from the arm of the X is called the dorsal root. The ventral root is on the belly side, and the dorsal root is on the back side. The dorsal root ganglion is a bulge halfway between where the nerve leaves the spine and where the dorsal and ventral roots join. Sensory neuron somas cluster in the dorsal root. Motor neuron somas cluster in the gray matter in the leg of the X-motor neuron axons are bundled in the ventral root. Figure 35. 29 spinal nerves contain both sensory and motor axons. The somas of sensory neurons are located in dorsal root ganglia. The somas of motor neurons are found in the ventral portion of the gray matter of the spinal cord. Learning objectives. By the end of this section, you will be able to do the following. Describe the symptoms, potential causes, and treatment of several examples of nervous system disorders. A nervous system that functions correctly is a fantastically complex, well-oiled machine. Synapses fire appropriately, muscles move when needed, memories are formed and stored, and emotions are well regulated. Unfortunately, each year millions of people in the United States deal with some sort of nervous system disorder. While scientists have discovered potential causes of many of these diseases and viable treatments for some, ongoing research seeks to find ways to better prevent and treat all of these disorders. Neurodegenerative disorders. Neurodegenerative disorders are illnesses characterized by a loss of nervous system functioning that are usually caused by neuronal death. These diseases generally worsen over time as more and more neurons die. The symptoms of a particular neurodegenerative disease are related to where in the nervous system the death of neurons occurs. Spinocerebellar ataxia, for example, leads to neuronal death in the cerebellum. The death of these neurons causes problems in balance and walking. Neurodegenerative disorders include Huntington's disease, amyotrophic lateral sclerosis, Alzheimer's disease and other types of dementia disorders, and Parkinson's disease. Here, Alzheimer's and Parkinson's disease will be discussed in more depth. Alzheimer's disease. Alzheimer's disease is the most common cause of dementia in the elderly. In 2012, an estimated 5.4 million Americans suffered from Alzheimer's disease, and payments for their care are estimated at $200 billion. Roughly 1 in every 8 people age 65 or older has the disease. Due to the aging of the baby boomer generation, there are projected to be as many as 13 million Alzheimer's patients in the United States in the year 2050. Symptoms of Alzheimer's disease include disruptive memory loss, confusion about time or place, difficulty planning or executing tasks, poor judgment, and personality changes. Problems smelling certain scents can also be indicative of Alzheimer's disease and may serve as an early warning sign. Many of these symptoms are also common in people who are aging normally, so it is the severity and longevity of the symptoms that determine whether a person is suffering from Alzheimer's. Alzheimer's disease was named for Alois Alzheimer, a German psychiatrist who published a report in 1911 about a woman who showed severe dementia symptoms. Along with his colleagues, he examined the woman's brain following her death and reported the presence of abnormal clumps, which are now called amyloid plaques, along with tangled brain fibers called neurofibrillary tangles. Amyloid plaques, neurofibrillary tangles, and an overall shrinking of brain volume are commonly seen in the brains of Alzheimer's patients. Loss of neurons in the hippocampus is especially severe in advanced Alzheimer's patients. Figure 35. 30 compares a normal brain to the brain of an Alzheimer's patient. Many research groups are examining the causes of these hallmarks of the disease. One form of the disease is usually caused by mutations in one of 3 known genes. This rare form of early-onset Alzheimer's disease affects fewer than 5% of patients with the disease and causes dementia beginning between the ages of 30 and 60. The more prevalent, late-onset form of the disease likely also has a genetic component. One particular gene, apolipoprotein E, APOE, has a variant E4 that increases a carrier's likelihood of getting the disease. Many other genes have been identified that might be involved in the pathology. Link to learning. Visit this website for video links discussing genetics and Alzheimer's disease. Unfortunately, there is no cure for Alzheimer's disease. Current treatments focus on managing the symptoms of the disease. Because decrease in the activity of cholinergic neurons that use the neurotransmitter acetylcholine, is common in Alzheimer's disease, Several drugs used to treat the disease work by increasing acetylcholine neurotransmission, often by inhibiting the enzyme that breaks down acetylcholine in the synaptic cleft. Other clinical interventions focus on behavioral therapies like psychotherapy, sensory therapy, and cognitive exercises. Since Alzheimer's disease appears to hijack the normal aging process, research into prevention is prevalent. Smoking, obesity, and cardiovascular problems may be risk factors for the disease, so treatments for those may also help to prevent Alzheimer's disease. Some studies have shown that people who remain intellectually active by playing games, reading, playing musical instruments, and being socially active in later life have a reduced risk of developing the disease. A cross-section of a normal brain and the brain of an Alzheimer's patient are compared. In the Alzheimer's brain, the cerebral cortex is greatly shrunken in size, as is the hippocampus. Ventricles, holes in the center and bottom right and left parts of the brain, are also enlarged. Figure 35. 30 compared to a normal brain, left. The brain from a patient with Alzheimer's disease, right, shows a dramatic neurodegeneration, particularly within the ventricles and hippocampus. Credit, modification of work by Garando/Wikimedia Commons based on original images by ADEAR. Alzheimer's Disease Education and Referral Center, a service of the National Institute on Aging. Parkinson's disease. Like Alzheimer's disease, Parkinson's disease is a neurodegenerative disease. It was first characterized by James Parkinson in 1817. Each year, 50,006,000 people in the United States are diagnosed with the disease. Parkinson's disease causes the loss of dopamine neurons in the substantia nigra, a midbrain structure that regulates movement. Loss of these neurons causes many symptoms including tremor, shaking of fingers or a limb, slowed movement, speech changes, balance and posture problems, and rigid muscles. The combination of these symptoms often causes a characteristic slow, hunched shuffling walk, illustrated in Figure 35. 31. Patients with Parkinson's disease can also exhibit psychological symptoms, such as dementia or emotional problems, although some patients have a form of the disease known to be caused by a single mutation. For most patients, the exact causes of Parkinson's disease remain unknown. The disease likely results from a combination of genetic and environmental factors, similar to Alzheimer's disease. Post-mortem analysis of brains from Parkinson's patients shows the presence of Lewy bodies, abnormal protein clumps, in dopaminergic neurons. The prevalence of these Lewy bodies often correlates with the severity of the disease. There is no cure for Parkinson's disease, and treatment is focused on easing symptoms. One of the most commonly prescribed drugs for Parkinson's is L-Dopa, which is a chemical that is converted into dopamine by neurons in the brain. This conversion increases the overall level of dopamine neurotransmission and can help compensate for the loss of dopaminergic neurons in the substantia nigra. Other drugs work by inhibiting the enzyme that breaks down dopamine. Illustration shows a hunched man with stiff arms and a shuffling walk. Figure 35. 31 Parkinson's patients often have a characteristic hunched walk. Neurodevelopmental disorders. Neurodevelopmental disorders occur when the development of the nervous system is disturbed. There are several different classes of neurodevelopmental disorders. Some, like Down syndrome, cause intellectual deficits. Others specifically affect communication, learning, or the motor system. Some disorders like autism spectrum disorder and attention deficit hyperactivity disorder have complex symptoms. Autism spectrum disorder, ASD, is a neurodevelopmental disorder. Its severity differs from person to person. Estimates for the prevalence of the disorder have changed rapidly in the past few decades. Current estimates suggest that 1 in 88 children will develop the disorder. ASD is 4 times more prevalent in males than females. Link to learning. This video discusses possible reasons why there has been a recent increase in the number of people diagnosed with autism. A characteristic symptom of ASD is impaired social skills. Children with autism may have difficulty making and maintaining eye contact and reading social cues. They also may have problems feeling empathy for others. Other symptoms of ASD include repetitive motor behaviors, such as rocking back and forth, preoccupation with specific subjects, strict adherence to certain rituals, and unusual language use. Up to 30% of patients with ASD develop epilepsy, and patients with some forms of the disorder, like Fragile X, also have intellectual disability. Because it is a spectrum disorder, other ASD patients are very functional and have good to excellent language skills. Many of these patients do not feel that they suffer from a disorder and instead think that their brains just process information differently. Except for some well-characterized, clearly genetic forms of autism, like Fragile X and Rett syndrome, the causes of ASD are largely unknown. Variants of several genes correlate with the presence of ASD, but for any given patient, many different mutations in different genes may be required for the disease to develop. At a general level, ASD is thought to be a disease of incorrect wiring. Accordingly, brains of some ASD patients lack the same level of synaptic pruning that occurs in non-affected people. In the 1990s, a research paper linked autism to a common vaccine given to children. This paper was retracted when it was discovered that the author falsified data, and follow-up studies showed no connection between vaccines and autism. Treatment for autism usually combines behavioral therapies and interventions, along with medications to treat other disorders common to people with autism, depression, anxiety, obsessive-compulsive disorder. Although early interventions can help mitigate the effects of the disease, there is currently no cure for ASD. Attention Deficit Hyperactivity Disorder, ADHD. Approximately 3 to 5% of children and adults are affected by Attention Deficit Hyperactivity Disorder, ADHD. Like ASD, ADHD is more prevalent in males than females. Symptoms of the disorder include inattention, lack of focus, executive functioning difficulties, impulsivity, and hyperactivity beyond what is characteristic of the normal developmental stage. Some patients do not have the hyperactive component of symptoms and are diagnosed with a subtype of ADHD, Attention Deficit Disorder, ADD. Many people with ADHD also show comorbidity in that they develop secondary disorders in addition to ADHD. Examples include depression or obsessive-compulsive disorder, OCD. Figure 35. 32 provides some statistics concerning comorbidity with ADHD. The cause of ADHD is unknown, although research points to a delay and dysfunction in the development of the prefrontal cortex and disturbances in neurotransmission. According to studies of twins, the disorder has a strong genetic component. There are several candidate genes that may contribute to the disorder, but no definitive links have been discovered. Environmental factors, including exposure to certain pesticides, may also contribute to the development of ADHD in some patients. Treatment for ADHD often involves behavioral therapies and the prescription of stimulant medications, which paradoxically cause a calming effect in these patients. Bar graph shows that 49% of ADHD patients suffer from ADHD alone. 7% have both ADHD and conduct disorder. 11% have ADHD and depression. 11% have ADHD and anxiety disorder. 24% have ADHD and a combination of depression, anxiety disorder, and conduct disorder. Figure 35. 32 Many people with ADHD have one or more other neurological disorders. Credit chart design and illustration. Modification of work by Lee Coriel. Credit data. Doctors. Biederman and Farion, Massachusetts General Hospital. Career connection. Neurologist. Neurologists are physicians who specialize in disorders of the nervous system. They diagnose and treat disorders such as epilepsy, stroke, dementia, nervous system injuries, Parkinson's disease, sleep disorders, and multiple sclerosis. Neurologists are medical doctors who have attended college, medical school, and completed 3 to 4 years of neurology residency. When examining a new patient, a neurologist takes a full medical history and performs a complete physical exam. The physical exam contains specific tasks that are used to determine what areas of the brain, spinal cord, or peripheral nervous system may be damaged. For example, to check whether the hypoglossal nerve is functioning correctly, the neurologist will ask the patient to move their tongue in different ways. If the patient does not have full control over tongue movements, then the hypoglossal nerve may be damaged, or there may be a lesion in the brainstem where the cell bodies of these neurons reside, or there could be damage to the tongue muscle itself. Neurologists have other tools besides a physical exam they can use to diagnose particular problems in the nervous system. If the patient has had a seizure, for example, the neurologist can use electroencephalography, EEG, which involves taping electrodes to the scalp to record brain activity, to try to determine which brain regions are involved in the seizure. In suspected stroke patients, a neurologist can use a computerized tomography, CT, scan, which is a type of X-ray, to look for bleeding in the brain or a possible brain tumor. To treat patients with neurological problems, neurologists can prescribe medications or refer the patient to a neurosurgeon for surgery. Link to learning. This website allows you to see the different tests a neurologist might use to see what regions of the nervous system may be damaged in a patient. Mental illnesses. Mental illnesses are nervous system disorders that result in problems with thinking, mood, or relating with other people. These disorders are severe enough to affect a person's quality of life and often make it difficult for people to perform the routine tasks of daily living. Debilitating mental disorders affect approximately 12.5 million Americans, about 1 in 17 people, at an annual cost of more than $300 billion. There are several types of mental disorders including schizophrenia, major depression, bipolar disorder, anxiety disorders and phobias, post-traumatic stress disorders, and obsessive-compulsive disorder, OCD, among others. The American Psychiatric Association publishes the Diagnostic and Statistical Manual of Mental Disorders, or DSM, which describes the symptoms required for a patient to be diagnosed with a particular mental disorder. Each newly released version of the DSM contains different symptoms and classifications as scientists learn more about these disorders, their causes, and how they relate to each other. A more detailed discussion of two mental illnesses, schizophrenia and major depression, is given below. Schizophrenia. Schizophrenia is a serious and often debilitating mental illness affecting 1% of people in the United States. Symptoms of the disease include the inability to differentiate between reality and imagination, inappropriate and unregulated emotional responses, difficulty thinking, and problems with social situations. People with schizophrenia can suffer from hallucinations and hear voices. They may also suffer from delusions. Patients also have so-called negative symptoms like a flattened emotional state, loss of pleasure, and loss of basic drives. Many schizophrenic patients are diagnosed in their late adolescence or early 20s. The development of schizophrenia is thought to involve malfunctioning dopaminergic neurons and may also involve problems with glutamate signaling. Treatment for the disease usually requires antipsychotic medications that work by blocking dopamine receptors and decreasing dopamine neurotransmission in the brain. This decrease in dopamine can cause Parkinson's disease-like symptoms in some patients. While some classes of antipsychotics can be quite effective at treating the disease, they are not a cure, and most patients must remain medicated for the rest of their lives. Depression. Major depression affects approximately 6–7% of the adults in the United States each year and is one of the most common mental disorders. To be diagnosed with major depressive disorder, a person must have experienced a severely depressed mood lasting longer than 2 weeks along with other symptoms, including a loss of enjoyment in activities that were previously enjoyed, changes in appetite and sleep schedules, difficulty concentrating, feelings of worthlessness, and suicidal thoughts. The exact causes of major depression are unknown and likely include both genetic and environmental risk factors. Some research supports the classic monoamine hypothesis, which suggests that depression is caused by a decrease in norepinephrine and serotonin neurotransmission. One argument against this hypothesis is the fact that some antidepressant medications cause an increase in norepinephrine and serotonin release within a few hours of beginning treatment. But clinical results of these medications are not seen until weeks later. This has led to alternative hypotheses. For example, dopamine may also be decreased in depressed patients, or it may actually be an increase in norepinephrine and serotonin that causes the disease, and antidepressants force a feedback loop that decreases this release. Treatments for depression include psychotherapy, electroconvulsive therapy, deep brain stimulation, and prescription medications. There are several classes of antidepressant medications that work through different mechanisms. For example, monoamine oxidase inhibitors, MAO inhibitors, block the enzyme that degrades many neurotransmitters, including dopamine, serotonin, norepinephrine, resulting in increased neurotransmitter in the synaptic cleft. Selective serotonin reuptake inhibitors, SSRIs, block the reuptake of serotonin into the presynaptic neuron. This blockage results in an increase in serotonin in the synaptic cleft. Other types of drugs, such as norepinephrine dopamine reuptake inhibitors and norepinephrine serotonin reuptake inhibitors, are also used to treat depression. Other neurological disorders. There are several other neurological disorders that cannot be easily placed in the above categories. These include chronic pain conditions, cancers of the nervous system, epilepsy disorders, and stroke. Epilepsy and stroke are discussed below. Epilepsy. Estimates suggest that up to 3% of people in the United States will be diagnosed with epilepsy in their lifetime. While there are several different types of epilepsy, all are characterized by recurrent seizures. Epilepsy itself can be a symptom of a brain injury, disease, or other illness. For example, people who have intellectual disability, or ASD, can experience seizures, presumably because the developmental wiring malfunctions that cause their disorders also put them at risk for epilepsy. For many patients, however, the cause of their epilepsy is never identified and is likely to be a combination of genetic and environmental factors. Often, seizures can be controlled with anticonvulsant medications. However, for very severe cases, patients may undergo brain surgery to remove the brain area where seizures originate. Stroke. A stroke results when blood fails to reach a portion of the brain for a long enough time to cause damage. Without the oxygen supplied by blood flow, neurons in this brain region die. This neuronal death can cause many different symptoms, depending on the brain area affected, including headache, muscle weakness or paralysis, speech disturbances, sensory problems, memory loss, and confusion. Stroke is often caused by blood clots and can also be caused by the bursting of a weak blood vessel. Strokes are extremely common and are the third most common cause of death in the United States. On average, one person experiences a stroke every 40 seconds in the United States. Approximately 75% of strokes occur in people older than 65. Risk factors for stroke include high blood pressure, diabetes, high cholesterol, and a family history of stroke. Smoking doubles the risk of stroke. Because a stroke is a medical emergency, patients with symptoms of a stroke should immediately go to the emergency room, where they can receive drugs that will dissolve any clot that may have formed. These drugs will not work if the stroke was caused by a burst blood vessel or if the stroke occurred more than 3 hours before arriving at the hospital. Treatment following a stroke can include blood pressure medication to prevent future strokes and sometimes intense physical therapy.